So welcome to this video where again we're looking at transport mechanisms for bile acids, and this time we're looking to see how we move these bile acids from the sinusoid across into the hepatocyte and then into the canaliculus. Before we start, we have to remember a couple of things. First, about 90 to 95% of your bile acids are recycled from the GI tract, which means they're going to be found in both their conjugated and unconjugated form. They're going to travel through the portal vein, make their way to the sinusoid, and then you need to transport these across into the hepatocyte. So there's a couple of different ways that we can transport them. Let me just get this laser pointer up so we can see. Now this here is a sodium co-transport mechanism, and what it does, it will bring the bile acids in. Here we have the sinusoidal space, this is the hepatocyte, and here is the canaliculus. And so what we're looking at doing is getting these bile acids, be they conjugated or unconjugated, across into the hepatocyte and all the way across and into the canaliculus. So this first mechanism, the sodium co-transport mechanism, uses the concentration gradient of sodium, so it's a secondary transport mechanism to get both conjugated bile acids and unconjugated bile acids into the hepatocyte. So we can get both types in. It seems to favour conjugated, but unconjugated bile acids can also move into the hepatocyte this way. There's also an organic anion transporting polypeptide, OAT-P, that will transport bile acids into the hepatocyte. You can see unconjugated moving in, and as well as that, it will also move conjugated bile acids in. These don't require sodium co-transport and don't seem to require ATP, and their precise mechanism, we're still trying to work out what's going on here. So what we can now see is we have in the hepatocyte, we have both conjugated and unconjugated bile acids, and we need to get them from this area across to the canaliculus to be transported. Now, an important thing to understand is, however, is that only conjugated bile acids can make their way into the canaliculus, which means we have to conjugate these unconjugated bile acids. So the unconjugated bile acids are conjugated in the hepatocyte, and then these conjugated bile acids are shuttled across to the canaliculus with the help of what are called bile acid binding proteins. So they'll move these conjugated bile acids to be ready for transport at the canaliculus. Now, an important fact to look at before we talk about the transport mechanisms from the hepatocyte into the canaliculus is that the concentration of free conjugated bile acids inside the hepatocyte are about 1,000 times less than that of the canaliculus, which means that we're moving from a very low concentration or relatively low concentration to a very high concentration, which means therefore we're going to have to use energy to get these conjugated bile acids into the canaliculus. There are two major transport mechanisms. Most of the conjugated bile acids are transported into the canaliculus via this thing called BSEP, bile salt export pump, and this is an ATP binding cassette protein. So it uses ATP to move conjugated bile acids against their concentration gradient into the canaliculus. Now this isn't the only mechanism. We also have a secondary mechanism here, which is via MRP2 or multi-drug resistant protein. This too is an ATP binding cassette protein, and it too will move these conjugated bile acids against their concentration gradient into the canaliculus. So what you can see here now is that we've moved the conjugate or the bile acids, both unconjugated and conjugated, across the hepatocyte to the canaliculus, and they've now been transported into the canaliculus in their conjugated form against their concentration gradient. Once conjugated, the bile acids find it difficult to get back into the hepatocyte because they can't diffuse across plasma membranes, so therefore they're kept within the high concentration in the canalicular fluid. Now what happens once they're in the canalicular fluid is what we'll look at in another video.